endocrinology, receptors are proteins. Several classes of peptide hormone receptors have been defined. For example, the insulin receptor is a heterotetramer linked by multiple disulfide bonds in which the extracellular alpha subunit binds insulin and the membrane spanning beta subunit transduces the signal through the tyrosine protein kinase domain located in the cytoplasmic portion of the polypeptide. Receptors on the steroid or thyroid type are membrane of the large superfamily of the nuclear receptors. Many related members of this family currently have no known legend and thus are called orphan receptors. Classification of hormones, chemical structures, and mechanism of action. Based upon chemical structure, classification of hormones by the chemical composition. Amino acid hormones, thyroxine, triiodothyronine, adrenaline, non-adrenaline, melatonin. Steroid hormones, cortisol, aldosterone, estradiol, androgen, progesterone, testosterone, active form of vitamin D3. Polypeptide hormones, antidiuretic hormone, oxytocin, insulin, pituitary trophic hormone, calcitonin, PRL, GH, and LPH. Classification based on mechanism of action. Hormones are classified into two broad groups based on the location of the receptor to which they bind and the signal used to mediate their action. Classification of hormones according to mechanism of action. Group 1. Hormones that bind to intracellular receptors, cytosolic or nucleus, solubility, lipophilic, chemical composition derived from the cholesterol except thyroid hormones and retinoic acid. Transport in blood associated with the transport protein to tackle solubility problems, long plasma half-life. Mechanism of action. Free hormones traverses plasma membrane of the cell and binds to the receptor in the cytosol or nucleus of the target cell. Hormone receptor complex bind to the specific region in the DNA of the gene. Binding causes activation or inactivation of the specific gene transcription. Accordingly, amount of protein synthesis is changed. Accordingly, certain metabolic processes are affected. Group 1 hormones. Hormones in the group 1 are lipophilic. After secretion, these hormones associate with the plasma transport or carrier protein, a process that circumvents the problem of the solubility while prolonging the plasma half-life of the hormone. The bound form merely acts as a circulating reservoir, releasing the free hormone in the immediate vicinity of the target tissue as per cellular requirements. The relative percentage of the bound in the free hormone are determined by the amount, binding affinity, and binding capacity of the transport protein. Therefore, the ratio of free bound forms and not the total circulating hormone concentration is true reflection of the observed biological activity. Group 1 hormones include androgen, calcitriol, estrogen, glucocorticoids, mineralocorticoids, retinoic acid, and the thyroid hormones. Group 2 hormones. It consists of water-soluble hormones. They bind to specific receptors spanning the plasma membrane of the target cell. Hormones that bind to the surface receptor of the cell communicate with intracellular metabolic processes through intermediary molecules called second messengers, which are generated as the consequence of the ligand receptor interactions. Group 2 hormones are further subclassified based on the second messenger they generate. Second messenger including cyclic AMP, cyclic GMP, calcium kinase. Hormones that act through the cyclic AMP by stimulating beta androgenic, adrenergic, catecholamines, adrenocorticotropic hormones, calcitonin, chorionic, corticotropin releasing hormone, CRH, FSH, glucagon, LH, PH, TSH. by inhibiting cyclic AMP, A2 adrenergic, cerecholamine, somatostatin, and angiotensin 2.